we discovered how to violently rewrite the code of life, not in a lab, but by accidentally spilling our industrial poisons from the man-made world directly into nature's genetic blueprint. A fire at a Swiss chemical plant in 1986 sent 30 tons of pesticides and mercury into the Rhine River. The water turned blood red, killing an estimated 500,000 fish almost instantly. Surviving eels and other organisms later showed significant DNA damage and cellular abnormalities from the toxic brew. When the Exxon Valdez tanker split open in 1989, it coated Alaska's Prince William Sound in 11 million gallons of crude oil. Scientists later found that Pacific herring exposed to the oil developed genetic mutations. These changes caused heart defects and spinal deformities that were passed down to subsequent generations. The neighborhood of Love Canal was unknowingly built on 21,000 tons of buried toxic waste in Niagara Falls. Leaking chemicals, including the potent mutagen dioxin, seeped into homes and schools throughout the 1970s. Studies of residents showed a significantly higher frequency of chromosome damage compared to unexposed populations. In 2005, explosions at a Chinese petrochemical plant released 100 tons of benzene and other carcinogens into the Songhua River. The toxic slick stretched for 80 kilometers, contaminating the water for millions of people. Researchers studying fish in the aftermath discovered widespread DNA strand breaks and damage to their cellular structure. The Seveso disaster in Italy released a dense cloud of TCDD, the most potent form of dioxin, over a populated area in 1976. The long-term genetic impact was startling and specific. Men who were children during the exposure later fathered nearly twice as many daughters as sons, indicating the toxin had altered their reproductive cells. The 2010 Deepwater Horizon oil spill was made worse by the 2 million gallons of chemical dispersant used to clean it up. This toxic cocktail proved to be a powerful mutagen, causing widespread DNA damage in crabs, dolphins, and birds. Gulf killifish exposed to the chemicals underwent rapid genetic evolution just to survive in the poisoned waters. The widespread spraying of Agent Orange during the Vietnam War unleashed a potent contaminant known as dioxin. This chemical is a teratogen, a substance that directly interferes with fetal development. Its presence has been linked to horrific birth defects and genetic abnormalities spanning at least 3 generations in parts of Vietnam. For decades, a factory in Minamata, Japan, discharged methylmercury directly into the bay, contaminating the entire food chain. The toxin caused catastrophic neurological damage and is known for causing congenital Minamata disease. Pregnant women who ate the fish gave birth to children with severe birth defects, a direct result of the chemical's interference with developing DNA. The 2011 Fukushima Daiichi disaster released radioactive isotopes that act as powerful mutagens by directly shattering DNA. The most famous example is the pale grass blue butterfly, which developed smaller wings, damaged eyes, and malformed legs. These mutations became more severe and more common in each new generation exposed to the radiation. The Chernobyl nuclear disaster in 1986 remains the most terrifying example of a chemical release altering an entire ecosystem's genetics. The intense radiation caused immediate widespread DNA damage in every living thing it touched. Birds in the zone were found with smaller brains. Some frogs turned black as a genetic adaptation to radiation, and the fallout is directly linked to increased rates of thyroid cancer in humans. These events are a stark reminder of how fragile our genetic code is in the face of industrial chemistry. Let us know in the comments which one of these cases you found the most unsettling.